This video is about introducing a spatio-temporal filtering in laser Doppler holography. The motivation for this work was to improve the quality of blood flow movies at low frequency. As when we showed these images to clinicians, they told us it was nice, but it would be better to be able to reveal the smallest blood vessels. And for us, it was not a problem of spatial resolution. The instrument has the capability to resolve smaller retinal features. It was a problem of flow resolution, and that meant that it would be very problematic to be able to be clinically relevant, as we would not be able to reveal blood flow in pathological vessels where it is slowed down. And it was very clear when we were comparing LDH blood flow images with those provided by OCT angiography, which here sort of serves as ground truth. There are many smaller blood vessels that we miss with laser Doppler holography for a lack of sensitivity to slower blood flow, and you can see all of these vessels with OCTA that we cannot see with LDH. So to introduce the problem, to show you that it's a problem of flow resolution, and to present you the solution that we implemented, I need to go back to the basics of laser Doppler holography. So we have this eye that we illuminate with a single frequency laser diode, and we collect the Doppler broadened beam that we combine on the reference arm with the reference arm on the camera. And on the camera we're able to measure the Doppler spectrum. Now, there are multiple contributions to the Doppler broadening, and we're only interested in blood flow. So there is the light scattering on red cells that flow at very high speed in blood vessels. That is what we're interested in, but there's also a Doppler broadening that is induced by the whole motion of the eye, so the fixational eye movement and the pulsatile axial motion of the eye, and that's what we are not interested in. And of course we use a Fourier transform at first to analyze this Doppler spectrum, because we're looking into frequency shifts, so a Fourier transform is very natural to do so. We have this full hologram stack of a few seconds of recording. We slide a short time window along the stack. We calculate the Fourier transform at its position in time, and by integrating the highest frequency, the signal at high frequency that we call power Doppler of this Fourier transform, we're able to have a metric, a quantity that is related to blood flow. So more energy at high frequency means more light that has been scattered by red cells, so more blood flow. So to do so, we have a signal that is sampled at 70 kilohertz or so, and we have a short time window of a few hundred images, which gives a resolution of a few milliseconds. Now, the problem with the Fourier transform is that we need to make a compromise. When we integrate the power Doppler, we have a frequency threshold, a lower cutoff frequency. And if we look at the low frequency power Doppler, for example 1 to 5 kilohertz, we have something that is able to reveal very nicely the smallest blood vessels, because small blood vessels mean slower blood flow, so lower frequency shift, and it's visible at low frequency. But on the other hand, it's a signal that is completely dominated by eye motion. If you look at the temporal dynamic of the signal, the plot is very messy. And at very high frequency, it's the opposite. You have a very nice pulse waveform that is impeccable, but the image is not able to reveal the smallest blood vessels. So there's a compromise we need to make between how much we want the image to reveal smaller features and the quality of the dynamic signal that we will measure. And here is the source of the problem. In an ideal case, if you look at the Fourier transform, the temporal Fourier transform of the signal, you have at high frequency the contribution of blood flow, at low frequency the contribution of eye motion, and they are nicely spectrally separated. So in the Fourier domain they are separated, and you can just integrate nicely blood flow without having contribution of eye motion. But of course, in reality, whether you're looking at slower blood flow or in case the eye motion are stronger than usual, well, there is an overlap of the two parts of the spectrum, and where the two contributions are overlapped, the signal of blood flow is corrupted and it cannot be retrieved. And what this means is that a Fourier decomposition, the Fourier space is not adequate to separate blood flow from eye motion. So luckily, this is a very common problem in medical imaging. It has been met elsewhere, and especially in acoustic Doppler imaging. And so we implemented a solution that was found for ultrasound Doppler imaging, and it's very remarkable how the context, the problem, the solution are very similar in optical and acoustic Doppler imaging. So this is a very nice paper that explains things very well. So the solution that was proposed in that paper is, instead of doing a Fourier transform to a singular value decomposition where it's no longer a temporal transformation, it's a new space, a new decomposition that takes into account both space and time. So we have a short-term window of anti-holograms that is reshaped into a 2D matrix. So on one dimension there is time, on the other dimension there is space. So now the column of the new matrix is our images, and this new matrix is decomposed with singular value decomposition into a product of 3 matrices. So in the matrix in the middle, these are the singular values, and each of the singular value is associated to one spatial singular vector and one temporal singular vector. And now the original matrix is written as the summation of terms, and each of these terms is determined by the product of a temporal pattern, a 1D temporal pattern, by an image, which is the spatial singular vector, and that is weighted by a singular value. And this allows contributions that have a very specific spatial pattern to be isolated in singular value. So it decomposes and isolates the singularities. So this is one way of representing the singular value decomposition. On the plot on the left, you have the energy of the ordered singular values. So there's close to 100 decibels between the first and last singular values. On the middle graph, you have a representation of the spectral energy of the temporal singular vectors. So what you can understand is that the first singular vectors, they have very slow variation, slow temporal variation, and the last temporal singular vectors, they have faster variation. And finally, the last graph shows the correlation matrix of the spatial singular vectors, and what you can see is that there's sort of 2 subspaces that differentiate. So one subspace that we will see afterwards is associated to clutter, because all sorts of eye motion, specular reflection, they have a very high spatial coherence, so they are associated to singular values of high energy, and all the rest is blood flow, and all of the images reveal blood flow, so they are correlated among one another. So here is an example of single value decomposition. We're looking at the spatial eigenvectors. So those associated to singular values of very high energy, they look something like this. And those associated to singular values of lower energy, they look something like this. And what this means is that the first singular values, they reveal clutter, something like eye motion, specular reflection on cornea or other surfaces. And the higher values are able to reveal blood flow. And the reason of that is that something like eye motion is going to generate the same Doppler frequency shift over the whole field of view. So each pixel of the field of view has a contribution that is similar to all the other pixels. And when you do a singular value decomposition, this is isolated into a singularity, into a specific singular value. And this has high energy because it's present over the whole field of view. And in contrast, something like blood flow, it's contribution of completely uncorrelated scattering events. So it's Doppler contributions that are not correlated over the whole field of view. So that's why they are isolated, well, in the singular values of lower energy. So what this means is that the singular value decomposition is much more powerful to separate clutter from blood flow than the Fourier transform. The Fourier transform was only taking into account the temporal dynamics of the signal, and now the SVD takes into account both the temporal and spatial aspects of the signal. So in practice, the SVD filtering is implementing the following way. We have our filtered matrix where we just remove the first singular vectors because it's assumed that they are associated to clutter. So all the singular values of highest energy are removed, and the trick is just to set the right threshold. And then we do the usual Fourier analysis. And the advantage of an SVD compared to a PCA is that it allows you to remain in the normal domain. So you can do a Fourier transform and then be able to have a signal that is related to kilohertz and velocities in the end. Right, so here is examples of images that are very much affected by specular reflection. So it could be corneal reflection or reflection. And the third example is reflection on the beam splitter cube. And you can see that at low frequency, so 2 to 6 kilohertz is low frequency for us, the Fourier transform is not able to separate blood flow from corneal reflection because they are in the same frequency range. And when we do the prior SVD filtering and then the Fourier analysis, we're able to significantly increase the contrast of the blood flow images. We can do pretty much the same with eye motion. So for the same reason, eye motion is something very correlated over the whole field of view, like corneal reflection. And when we do the prior SVD filtering before doing the Fourier analysis, we're able to largely increase the signal of the image. And to our surprise, it was also very powerful to increase the contrast of images that were affected by camera jitter. So we have this ultra-fast camera, and when we leave it on for a few hours, then the quality of the images starts to decrease, and they were affected by this sort of rectangular pattern. And unlike the other example, this contribution is at very high frequency. So usually we were able to remove eye motion by just looking at the very high frequencies, but this time we were facing a problem that was located at high frequency. And with the singular value decomposition, we removed it, and that is because it's a signal that is very well temporally localized. It's a specific pattern at a specific temporal frequency, and when it's affecting many pixels, then it's also isolated into a singular value. So that's why it was also able to increase the contrast of images. So the purpose of this slide is to show what happens with the SVD filtering throughout cardiac cycle when there is no eye motion. So first, in presence of mild eye motion that we define as motion that will generate a Doppler contribution that is lower than the integration threshold. So here the power Doppler was integrating above 2 kilohertz and the eye movement was located at something like 1 kilohertz. And what you see is that with the power spectrum that represent the SVD filtered signal, the eye motion is removed, but it's below the power Doppler integration threshold. So the two curves with and without SVD filtering are exactly alike above the power Doppler threshold. So when we make the images, they are close to identical. But now in presence of strong motion, where we have an eye motion that generates a Doppler contribution above the power Doppler integration threshold, well, what you see is that the SVD is able to remove this Doppler contribution, and now the power Doppler is going to be different because the two curves above the integration threshold are different, and the image without SVD filtering is completely dominated by eye motion, and the image filtered by SVD is able to reveal blood flow. So what happens is that the power Doppler in presence of eye motion is dominated by eye motion, and the contribution of blood flow is very tiny, so that's why the contrast is very poor. And if we look at the spectrogram of the signal variation with and without SVD, you see in red the contribution of eye movement, so it's something that is pulsatile and it's removed by the SVD. So that's why every time there is eye movement, it's removed by the SVD, and without the SVD we're not able to measure the nice clean waveform, and with the SVD we're able to measure something that reveals the arterial profile. So what this means is that the SVD is critical to measure blood flow in smaller vessels, because otherwise the dynamic of the signal is completely dominated by eye motion, and SVD gives the access to monitoring blood flow in smaller blood vessels. And this is what it amounts to when we look at blood flow movies. On the left is just with Fourier filtering, and on the right is SVD plus Fourier. And on the left there is all of this pulsatile eye motion, and the dynamic is very poor, and on the right it's very nicely revealing all the blood flow at low frequency. And this ability to reveal blood flow at low frequency is absolutely crucial because it's the smaller blood vessels, and it's also choroidal veins. So in this example we're combining the movie on the left, which is the 2 to 6 kilohertz in cyan, and the high frequency 6 to 30 kilohertz in red on the movie on the right. And before we were not able to do that because the movie on the left was completely dominated by eye motion. And this was particularly helpful for clinical cases. So here it's a case of retinal vein occlusion. You have the artery that fits the territory of the vein, where you have a great pulsatility that is revealed nicely, but the vein cannot be revealed with the high frequency. And then, thanks to the SVD, as we can access the 2 to 6 kilohertz frequency range, we're able to reveal the vein that here appears in blue. So that is why the SVD has been absolutely critical for the clinical relevance of the instrument, because otherwise we would not be able to really image blood flow in pathologies. It has been completely critical also to study the choroid, because as I explained in another presentation, choroidal arteries are revealed by high frequencies because they have higher flow, so we didn't really need the SVD to reveal them because high frequencies are anyway free of eye movement. But choroidal veins, they have slower blood flow, so they are revealed by lower frequencies, and for this it was absolutely critical to have a clean signal at low frequencies. So these are the montage that we were able to make of the choroid with the SVD filtering, and it brings a lot of information compared to ICG and OCT. So in conclusion, we demonstrated that the spatio-temporal decomposition is much more powerful than Fourier transform to be able to separate blood flow from eye movement. So the SVD is just dramatically increased the contrast of blood flow images, and this access to low frequency is crucial to reveal, well, the smaller blood vessel where blood flow is slower. It's critical to reveal blood flow fluctuations that are free of eye movements. It's important to be able to image the choroid because it reveals choroidal veins, and it's indispensable to reveal blood flow in pathology. So this SVD filtering is just a few lines of code, but it has been extremely important for the clinical relevance of the instrument.